Thank you for attending today's RDA webinar on biosharing recommendations, data repositories, standards and policies in the life, biomedical and environmental sciences. I am Katrin Beck from the RDA Europe Training Program team. Now let me present you our speaker, Dr. Peter McQuilton. Peter McQuilton is the project coordinator for the biosharing project, based at the University of Oxford eResearch Centre in UK. He holds a PhD in Drosophila embryonic nervous system development from the University of Cambridge. Prior to working on the biosharing project, he worked for over 10 years as a biocurator at Flybase, a model organism database focused on the genetics and genomics of the fruit fly. Peter has been heavily involved in the standardization and promotion of biocuration, and his work in this area has culminated in election to the International Society of Biocuration Executive Committee. As biosharing project coordinator, Peter's activities are in and around data curation, text mining, ontology design, and data sharing and publication in the life, natural, and biomedical sciences. So let's start our webinar. Peter?
Okay, thank you. Hopefully everyone can hear me. Unless you start shouting at me, I'd assume so. I've also started my webcam so you can see my beautiful face. Okay, off we go. Okay, so thank you very much, Catherine, for that introduction. So over the next 30 minutes or so, I'll be talking to you about our work in the biosharing working group over the last 18 months to map and beyond, to map the landscape of data standards, data repositories, and data policies, in particular using our use case of the life sciences, which includes the biomedical sciences, agricultural sciences, and environmental sciences. I'll be doing that on behalf of the joint RDA Force11 BioSharing Working Group and my co-chairs, in particular Susanna Sundazansone, Sam Hodgson, and Rebecca Lawrence. And I'm coming to you from the Oxford eResearch Centre at the University of Oxford in the UK, and just to say that biosharing is part of Elixir UK, if that means anything to you. Okay. So what were the aims of the RDA Force11 working group? Well, the first one was to develop a set of guidelines or principles for linking information on databases, content standards, and journal and funder data policies in the life sciences. And once we've developed those guidelines, then to put them in action to develop a curated registry, building upon one that has been running since 2011, with the idea that we wanted to access and cross-search this information, such that a variety of stakeholders can make decisions on which standards or databases they should use in their research or should endorse for others to use. That registry can now be seen at the website there, which is www.biosharing.org. You can see our work there, and that's what I'll be talking about today. But just to give you a sneak preview, that will be changing to fairsharing.org over the summer as we expand, building on our work in the life sciences to expand to curate information on other standards and databases and data policies outside of the life sciences. Okay, so the working group has been running for 18 months now, and as such is drawing to a close. And as part of that process, we've generated a set of recommendations, which can be found here in this recommendation package. Now, if I click here, I should have a little pointer, and I can point to you to this DOI for our recommendation package, where if you take a note of that, write that down, or look on the RDA website, find it, you can see we've produced a document which includes our set of guidelines and also more information about the registry. Okay, and do take a look at that document when you can, and leave any comments, as it's currently available for community review. And the more comments you leave, ultimately the better the guidelines and the registry will be. So please, I encourage you to have a look, read through, and leave any comments you can. Okay, there we go. So what is biosharing now then? Well, it is a web-based, manually curated and searchable portal that monitors the use and development and evolution of data standards, their use in databases or data repositories, and the adoption of both of these in data policies, be they from journals or funders. And our aim really with biosharing is to inform and educate a variety of different users. So we're trying to map a complex and constantly evolving landscape. And here you can see on the top left, I've got the data policies, top right, databases, and then in the center, I have the content standards, which is split into three. And these descriptors are essential for the interpretation, verification, and reproducibility or sharing of data sets. And they're often considered the invisible guiding hand underneath bioinformatics. And what we're trying to do is capture more information about them and relate them to everything else that people know about in order to surface this kind of information. So here we split them into three. We've got the formats, terminologies, and guidelines. So formats, or models and formats, are conceptual schema or exchange formats, really. They're a way of representing data or moving data between different databases. An example of one such format would be the FASTA format, which I believe started back in 1983 and is used for the sequence of nucleotide or protein sequencing. And then we have the terminologies, which you may know as control vocabularies or taxonomies, thesauri. I know them as ontologies, and the best example of these would be the gene ontologies, capturing information on biological process, molecular function, or cellular components in the life sciences. These are used to provide a structured hierarchical way of capturing information about a thing in a consistent manner. And then we have the guidelines or minimum reporting requirements. These are checklists of the basic information or metadata one would need to associate with an experiment in order to place that experimental data into context. Information about what equipment was used, what techniques, what concentrations, and so on, in order to make that data fair. So where do these standards come from? Well, they can come from a number of places. Some of them come from standard organizations. These are groups that come together to define standards for a particular domain or organization. An example would be the ISO standards or the CDISC standards for clinical research. And then we also have standards coming from more community-driven groups, grassroots groups, such as the TADWIG group for biodiversity standards, which I'll talk a little bit about later, and the combined group for computational biology standards. So these standards don't just exist in isolation, they also relate to each other and to other objects. So for example, a reporting guideline may stipulate the use of a particular terminology or a particular format. And in biosharing, we capture that information of those relationships in order to place all this information into context. So here you can see I've highlighted that with the blue arrows going between the standards. In addition, these standards are used, implemented, and or curated by databases. And so we capture that information, which databases use which standards. So if you're a researcher at the start of your work and you're thinking, okay, I'm working on this particular type of data and I want to eventually deposit it in this data repository, then you can use biosharing to look at that area and go, okay, for proteomics, for example, I need to put my data into this database in this format. And so we can provide all that information. And what's more, to complete the triangle, many journals and funders also prescribe requirements for where people should deposit their data and in what format, kind of a recommended data repository list, for example. And so we capture that information too. The majority of the time, these data policies stipulate where one should put the data, but sometimes now they're also suggesting in what format one should use. But even if they just stipulate where one should put the data, implicitly they're also suggesting what format it should be, because you have to use a format that that data repository takes. Okay, so that's the kind of information we're capturing about this complex and rapidly evolving landscape. Just to give you an idea of the magnitude of the data that we're talking about, here you can see on this slide we've got the content standards represented in numbers. So we've got formats, terminologies, and guidelines again. And then you've got some examples down the bottom underneath there. And then you can see a simple bar chart telling you how many we know of in each type. So we have approximately 225 models or formats in biosharing. We don't know of any more really. These are formats that are either used solely in biology or are more generic formats that have some application in biology, such as XML, for example. And then in the center we have the terminologies or ontologies, and there we know of at least 554 in biology, and they're the sources BioPortal, which I'm sure you're all aware of. And then we have all the reporting guidelines, a much smaller set of 115, which we have in BioSharing. And these statistics are as of about a week ago, so May 2017. Okay, so I've mentioned a little bit about how standards can be related to databases, and it's actually a very complex relationship, and it's not very easy to make true assumptions or to do any true predictions. And I want to show you an example of why that's the case. So on this slide here, you can see on the left-hand side, we have two very well-known databases. We have GEO, the Gene Expression Omnibus, and ArrayExpress. Both deal with gene expression data. They both use the Miami reporting guideline for minimum information about a microarray experiment. So that's great, they're using the same guideline. Perhaps they're using other standards the same as well. Well, that's not quite the case, because in GEO, they also use two formats, the soft format and the minimal format, while in ArrayExpress, they've opted initially for the MageML format, and now that's been deprecated, they use the MageTab format. So there you can see they share some standards but not others. And then finally, because in ArrayExpress they're using the MageTab format, that stipulates the use of the EFO, experimental factor ontology terminology. So you can see between these two databases that use the same standard initially as a reporting guideline, they actually use very different formats and terminologies. So it's important that we accurately capture what's out there, often unfortunately manually, in order to make it as useful as possible, as there's no kind of hard and fast way of assuming or predicting this kind of data. Okay, so just to refocus us, why are we doing all this again? It's to help users make the right decision. So on this slide, you can see I've got 4 different types of user, 4 different stakeholders. On the top left, we have the researchers, where they can ask questions such as the one stated here. My funder's data policy recommends the use of established standards, but which are the established standards that are widely endorsed and applicable to my particular type of data? They can find that information out on BioSharing. Then we have the funders and journal editors who can recommend or endorse the use of particular standards or databases. So they will need to know which are the mature standards and which are the databases that are standard compliant that they should recommend to their authors. So they will need to have some indicators of metrics, which are those that have been adopted by the community, are well maintained, which standards are implemented in well-funded databases and so on. So they need to be able to get a handle on that kind of information. Then we have curators of databases and standard developers themselves, who may want to know if they're using the most up-to-date version of a terminology to annotate their data, or indeed if they want to know if they want to annotate a new type of data, what terminology to use, what's out there. Perhaps they can work with other people in a particular area to make a new format or standard. Then they can use BioSharing to see what's out there. And then finally we have librarians and data managers who often have people coming to them going, well, I found this bit of data from 10 years ago down the back of the sofa. I need to put it somewhere. Where do I put it? In what format should I put it in now? It's in a format which has long since been deprecated. What format should I use now, and where should I put it? So those data managers may not have domain-specific knowledge, and they can come to BioSharing to find that out and to find exactly what formats they should use and which data repositories they should be pointed towards. So those are the kind of questions we're trying to answer with all of this. Now this is the current biosharing.org homepage, where you go to the website. This is what you would see. I'm not going to take you through it all. I'm just wanting to highlight a few sections. In the central section, you can see we have in the darker blue our search bar. Here on the left-hand side, we have a simple search using a Solar index that does a free text search across all of the records in BioSharing, and can start typing things in there, and away you go. If you want to make a more precise search, you can use our advanced field by field search, and this allows you to engage our autocomplete. So you can start typing in a field, for example, in our domain field for particular domains or areas of biology, and then it will highlight those that are available and make a more precise search for you. And then you can refine all these searches down using our filter matrix, that allow you to search by a country or a particular funder and so on. And I'll show you a little bit more about that later. If you're not entirely sure what you want to search for and you want a little bit of hand-holding through the search, you can use our new search wizard, Ask BioSharing. Now, this is currently quite embryonic, but it still allows you to search by domain and species, and over the summer we'll be adding other options as well, such as via licensing for a standard or a database, or via recommendations and so on. But I'll talk a little bit more about that later. What I did want to highlight was just down the bottom here. You can see the three types of registry we have: standards, databases, and policies, and then the numbers involved. So in BioSharing currently, we have just under 700 data standards, over 800 databases, and over 90 data policies from funders and journals. These are being added to all the time, but that just gives you a little handle on the types of numbers that we're talking about in biology. Okay, so now I'll quickly take you through an example record. I won't go through everything, but I'll just highlight a few of the main features. So in the top left, as on all of our records, we have the name of the record, the abbreviation where appropriate, as many standards, for instance, are known more by their abbreviation than by the full name. And then we have this R symbol there. Now this is a status indicator for that resource. R in this case indicates ready to use. So we have a resource here which is well maintained, actively in use by the community, so there should be no issues for you using it. We have a number of other markers that I'll go on to talk about later. Underneath that, we have our general information section. This is common to all of the resources in BioSharing. You can see at the top there we have a free text curators section just explaining in brief what the resource is about. Then we have the home page for that resource, where it was developed, when it was created, and what species it pertains to, if any, and what data types or the scope of the resource is appropriate. So here we have, it's a computational model, it's used in life science, it's used for network interaction models, and then underneath here we have what collections or recommendations this standard in this case is used in. I'll talk a little bit about those later, but these are essentially ways of collating the data for a particular domain, organization, or type. And then underneath there you can see each record in BioSharing. We allow you, if you are the owner of that resource, to maintain the record for that resource in BioSharing. So this is maintained by S. Keating, who is one of the people who have worked on the SBML standard. So they can, as a maintainer, they can now edit this record to ensure it's as up-to-date and accurate as possible. And when they do so, the BioSharing knowledge engineers receive notification, and then we can check that those edits conform to our curation guidelines, and vice versa. If we were to edit this record or perhaps make a new relationship to this record from another record, then the maintainer of this record receives an email notification that allows them to see what changes we've made and essentially do a cross-validation check to ensure we haven't made any mistakes anyway. And then underneath there we have a little timestamp that shows you when this record was added to BioSharing and when it was last updated, so you can see how recent the record is. If we were to scroll down the page, you see we have a number of other sections. I'm not going to go through all these, but I'll just highlight the one on the right hand side here that shows you the related standards and the implementing databases for this standard. So you can browse through here and browse through the data in BioSharing using this to click through. On the left, you can see a support section where we highlight if there's help available, what type of help, if there's a mailing list, Twitter and so on, are there any contacts. And I just want to highlight in here, we're also adding via our API a training section here, which I'll just show you on this slide, partnering with another Elixir UK resource, the TESS Training and Events portal. We're linking together training and events with the resources in BioSharing. So were you to look on TESS, for instance, here there's the metabolites database. You can see at the bottom there's a link to the record on BioSharing for metabolites. And then on the BioSharing record for metabolites, you can see there's a link across to the training. This is something that we're actively working on. We now have these simple links, but we're also using our API to share some of our general information section with the TESS website and also pull some information in from them on these training events. And then just below that, I just want to show this credit section again, where we can see the record is maintained. You can see who it's maintained by and find out more information about them. And then you can also see who's worked on it, what development group, and what funders have been involved, and where applicable, where we can find that information, what grant numbers have been involved. Okay, so I talked a little bit about how the landscape was constantly evolving and these indicators that we use for that. So you see the one at the top there, the ready for use indicator. We have another three. We have one which is in development. Now this is for resources, whether they're a database or a standard, where someone has started working on it. It's not quite ready for prime time, so it's not quite ready to accept data or to be used in a database, but they just wanted to make people aware that they were working in this area. So they can make a record on biosharing, label it as in development, and essentially use it as a way of advertising their new resource and asking for help. We then have a deprecated D indicator that we use for resources, mainly standards, that are no longer in use, as they've been subsumed in another ontology, for instance, or superseded by another reporting guideline. And where we have done that, where we have a resource which has been deprecated, we don't just leave you hanging. We explain in a note why it's been deprecated and also what other resource you should use instead. And we do that, as with everything in biosharing, by working with the community involved. So we actually go out and talk to the people who have made that resource, ensure that it is truly deprecated, and ask them where people should go instead. So the information is as accurate as possible. Okay. So I talked earlier about collections and recommendations, these groups of records. And so this is what I was talking about really. So collections group together one or more types of resource, so standards or databases, by domain, project, or organization. And the top three cards here are examples. So you see we can have the CDISC collection of the clinical research standards from the CDISC organization. We can have one for the combined standards. We have another here for the Tadwig biodiversity information standards. And then we have recommendations underneath there, which are a core set of resources that have been selected and recommended by a funder or journal data policy. You see we have a number there, the Ember Press, F1000 Research, and PLOS. And I should say again, as with everything in biosharing, these are created and curated with the people involved. So working with Tadwig, we've made their collection, and in this case, to show you the example, working with PLOS, we've made their recommendation. So if you were to click on that, this is what you would see. This is the PLOS recommendation at the top. You can see we have again some general information, hand curators with the PLOS data team. The record itself is maintained by PLOS, so by Emma from PLOS and other members of the PLOS data team. And then we have all the resources recommended by PLOS underneath. You can see just on the left-hand side, part of our filter matrix, where you can refine what you see here based on whether that resource has a publication out, whether it's ready for use, what country it's in, and so on, or refine it in various other ways. I've just highlighted here this little red recommended ribbon, which is found on any resource which is found in a recommendation. So when you do a search and you see a number of resources come up in your search list, and some of them have this red recommendation ribbon, that shows you that that resource has been recommended, and it acts as another indicator. If you have a number of resources to choose from, I would suggest you use one of those that has been recommended, as that shows community adoption and recommendation from data policy generators. One thing I want to show you now is our new graph viewer, Explore Biosharing. So all of our collections and recommendations now have this little kind of red orangey button, which shows the data in a more visually pleasing manner. So if I were to click on there, here you can see, hopefully you can see this in your screens, mine's quite small. So this is the PLOS recommendation. In the center here, you can see this is the PLOS data policy. And then coming out from that, you can see a number of resources. On the left-hand side here, you can see the legend, which gives you a breakdown of those resources. So PLOS recommends 61 databases. And also the relationships between those resources, so whether they're recommended, whether they are implementing particular standards and so on. And you might be able to see on your screen in grey a number of other relationships. Now these are links from these resources that have been recommended to other resources which are related to them. And this is quite useful if you want to use a recommendation as a jump off point to search through all the resources. So if I give you an example of that, here's Embo Press, the Embo Press recommendation. Here you can see we have a number of relationships between these objects, some standards here and databases. So standards are in yellow, databases in orange, and then you can see different relationships. So this purple relationship shows you that the database implements this standard. So this is a way of viewing the data. And then I can show you here, going back to the PLOS recommendation, browsing through, you can move the objects around. So here I've pulled together a number of different model organism databases, and I've seen that they all implement the gene ontology, but the gene ontology Go has not been recommended by PLOS. So here you can see this is where PLOS can look at this and go, well, perhaps since we're recommending the use of all of these databases, we are implicitly recommending the use of Go, and so we should make that more explicit in our data policy. So this is a way that people can use these recommendations and collections to actually see which standards they should or could be using. And indeed, I have a quote here from F1000 where they've stated that this browser will allow them to discover which databases and standards are not included in their author guidelines and should be, so allowing them to regularly monitor and refine their policies. Okay, if I now take you back to a collection, I'll show you the Tadwig collection, just to show you again a slightly different view. You can see here, these are all the Tadwig standards, and I've just got the RDA logo there to remind you that Tadwig is one of the, has a number of working groups in the RDA, and we're working with them now to represent their standards here in biosharing. So here you can see our work. These are all their data standards. And you can see the relationships between them. Some of these have evolved from each other. And so they are now thinking to just use this biosharing collection as a way of storing all of their data standards and the metadata surrounding them. Okay, so you can see I've used about 30 minutes of your time, so I'll start to wrap up. Just to say we work with a number of different communities, and we have a number of adopters throughout these communities. In the top left-hand side of this slide, you can see some of the societies and organisations that we work with, including FAIRDom, Force11, and the Eurobioimaging Group. And then we also work with various research data management services, such as the DMP tool, which also have a working group in the RDA, and various other institutional research data management services, such as those at Stanford, Oxford, and Cambridge. And then using our API, we're forging many crosslinks with other databases, including one with the National Bioscience Database Center in Japan, which is a collaboration which came out of our involvement with the RDA. And we're embedded in a number of projects and programs, including bioschemas and biocaddy. We work with a number of journal publishers, which you can see there, and with a number of standard developing groups. And just to highlight again and remind you, our recommendation is now out. This is the front page. You can see some of the people who have already adopted it. I encourage you once again to click on the link, take a look, leave a comment and adopt it if appropriate. And finally, I'd like to give some acknowledgements, because this is clearly not all my own work. Obviously, there's a team in Oxford led by Susana Santasansone, Philippe and Alejandra, the developers Milo and Massi, and Eamon, and then the knowledge engineers, myself, Ali, Delphine, and Melanie. There's our funders, and then on the right-hand side you can see our wonderful advisory board who advise us on many things and keep us on the straight and narrow. And with that, I'll thank you for your time. I can see there's a number of questions already in the chat, so I'll answer those first, and then I'll open it up for other comments. And if you do have comments or questions after the webinar, please do contact us on the email I've put on the screen there. BioSharing, contact us at list.sf.net. Okay, so thank you, and I'll start going through your questions.